Hello, everyone, and welcome to today's webinar, Advancing Therapeutic Antibody Development, Cutting-Edge Technologies for Faster Solutions. This webinar is part of the third annual event in the Immuno-Oncology Virtual Event Series. I am Israel Gonzalez, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, please visit www.genscript.com. Before we begin, I would like to remind everyone that this is an interactive event. We encourage you to first participate by communicating with the other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you'd like during the presentation. To do so, submit them into the Ask a Question box on your screen. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, you can also click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I now present today's speaker, Amanda Grimm, Senior Segment Marketing Manager, Antibody Drug Discovery at GenScript USA Incorporated. For a complete biography on our speaker today, please visit the Presenter tab from the menu on the left of your screen. Amanda, you may now begin your presentation.
Thank you for the introduction. It's great to be here to share different products and services for antibody drug development. The research being done by everyone here is so important to further understand cancer and find new treatments. We want to make sure scientists have resources to make their research easier and faster. So to get started, our contents of the presentation today will include a brief introduction to GenScript. We'll touch on some of the antibody history, and then we'll get into GenScript's antibody solutions, as well as what it would look like to work with us. So about GenScript, for those of you that may not be familiar with us, we have been around for over 20 years. Of those more than 20 years, over 18 have been dedicated to antibody service. And at the end of 2023, we had completed over 150,000 antibody projects. Here I have a slide showing our history and milestones of the GenScript group. We were founded in 2002 in New Jersey here in the US. Since then, we've added some additional subsidiaries like Legend and ProBio, our CDMO. A really huge milestone that I want to mention too was in 2022, our Legend group, in conjunction with Johnson & Johnson, received FDA approval for our first cell therapy product. And then here I just have a breakdown of the GenScript group, a little bit more information. I'm on the GenScript Life Science CRO side. I mentioned ProBio, our biologics CDMO. I touched on Legend, which is our cell therapy division. And then we also have Bestzyme, responsible for our synthetic biology. So we'll go ahead and we'll start with some antibody history. So what are antibody therapeutics? They're used to treat disease. The antibody is typically monoclonal and could be a whole molecule or a fragment of an antibody. Often these antibodies are modified to improve their function. Many that we're seeing right now are bi, tri, or multifunctional antibodies. These can be seen, in clinical trials. We're also seeing a lot of antibody drug conjugates. These are very highly investigated, and we're seeing a lot of the bigger companies invest in this technology. As far as the history, the first approval of a therapeutic antibody was in 1986. There's a little bit of, slow work, but a lot of research still happening in the antibody therapy space, what we see happening is by around 2006, we're starting to see more regular and annual approvals of antibody drugs. 2021 was a huge milestone for the 100th FDA-approved therapeutic, and that was our SARS-CoV-2 therapy treatment. So looking even more a little bit at the history, I do want to point out that over half of these therapeutic antibodies are treatments for cancer, the other half being non-cancer. This really shows that antibodies have demonstrated to be an effective method to treat cancer, and they continue to be as our different technologies are developed. And then here I compiled this chart of the top 20 antibody drugs. This is based on 2019 data, to further highlight the efficiency of antibodies as treatment, with half of these being treatments for various cancers like melanoma, breast cancer. The earliest approval listed is Rituxan back in 1997, and still is widely prescribed in its treatment for non-Hodgkin's lymphoma. So I do want to share a couple examples in the immuno-oncology space. The first is Keytruda, which is an example of a checkpoint inhibitor. This is manufactured by Merck and was approved in 2014. PD-1 is an immune checkpoint receptor that, when bound, turns off the T cell activation, and cancer cells take advantage of this by binding, thus quieting the normal T cell recognition that would normally eliminate these foreign cells. Keytruda comes in and it blocks PD-1 on T cell from binding that PD-L1 on the tumor cells that you can see in the picture on the bottom right. The T cells are now activated and the proper pathway can proceed to kill melanoma cells. And then my second example is Lunsumio. It is a bispecific antibody that is a T cell engager. It's developed by Genentech. It's still undergoing clinical studies. The intended use with this treatment of follicular lymphoma is for third line of treatment. What the antibody does here, by binding two epitopes, it brings together T cells via the CD3 to the tumor B cells via CD20. The goal with this then is to facilitate the immune system's ability to target and eliminate cancer cells. So we'll go ahead and, based on that information, we'll talk about GenScript solutions for antibody drug development. And to start off, here I have a high-level overview of the workflow. This workflow in particular is based on traditional hybridoma route just as a basis, but ultimately we've divided this into 4 development stages: target identification, lead gen and optimization, preclinical and clinical development. And then based on that workflow, we can expand here further into those 4 stages, and you can see a list of relevant products and services. The purpose of this is visual, just to see our coverage. Not all are covered in the slide deck. So if you don't see something or if you have questions, please don't hesitate to reach out. We are always looking for ways that we can help accelerate research. So we'll start with target identification. Like, say this is the first phase in the target discovery. We know that you're working diligently to identify the target, understand the associated disease mechanism, and there's various methods that this can be done, whether you're using, literature, maybe you already have known info from internal work that the lab has done. Maybe you're planning to conduct experiments. And more recently, AI and machine learning is allowing us to kind of circumvent some of this already by being able to model and then go directly to antibody development. For the purposes here, this is divided into 3 sections where GenScript offers products and services, that being gene analysis, protein analysis, and then cell-based analysis. So starting at the gene level, you may need to amplify your target using PCR and often to measure the DNA or RNA levels using qPCR or RT-qPCR. We offer ready-to-use reagents as well as offering our oligo service to create probes and primers specific to the project you're working on. Targets can be validated using gene editing to simulate disease condition or to correct it. GenScript has a variety of options for off-the-shelf CRISPR-Cas9 plasmids and enzymes to support the evaluation of target genes and functionality. We do offer functional guide RNA, and this is going to be generated specific to the customer's project. So the GeneTitan 8 million oligos, next-gen sequencing, and CRISPR guide RNA libraries enable comprehensive exploration of target genes, precise gene editing in order to gain deep insights into your gene's function. The GeneTitan 8 million oligos provide a pool of diverse sequences that can be used to identify your target and validate it. These are designed to cover a wide range of target genes, enabling that exploration of a broad landscape of potential targets. NGS allows for high throughput sequencing and analysis of genetic material. That information can then help you prioritize targets for further identification. CRISPR guide RNA libraries are a cutting-edge technology for efficient gene editing and functional genomics. We offer a comprehensive collection of guide RNA sequences that target specific genes or pathways of interest. One popular investigative disease is Alzheimer's, and so we do offer a library for that as an example. So for in-house analysis, we do offer gels and blotting automation as fundamental tools for protein detection and analysis. That includes the blotting units, membrane processing instruments, electrophoresis standards, and high-quality antibodies for screening. Automating Western work saves a lot of time from making the gels, uneven transfer, and a million tubes or trays for staining. We also offer a variety of immune checkpoint proteins and antibodies, which are going to be essential tools for further studying the immune system and developing your immunotherapies. GenScript's protein expression service covers multiple systems to accommodate the diverse needs and optimize production of target proteins. Efficient and reliable protein expression is crucial for obtaining sufficient quantities of those target proteins in order to characterize and further analyze functionality. For simple protein expression needs, the E. coli platform provides a cost-effective and rapid solution. This is ideal for the expression of soluble proteins, protein fragments, or small-scale production for that initial screening and characterization. The insect platform offers an alternative to the production of challenging proteins that require proper folding and maybe post-translational modifications. And then the mammalian platform offers that advantage of post-translational modifications and native folding. It's going to make it suitable for the production of the complex proteins and antibodies that may also require glycosylation. This system ensures generation of biologically active proteins that are going to more closely resemble their native counterparts. For target validation, frequently hundreds or even thousands of samples may need to be analyzed. This can be hard to do with Western. So ELISA kits are often used due to easy adaptation to high throughput and automation. By leveraging our expertise in antibody production and also assay development, we provide highly sensitive and specific ELISA kits tailored to your target proteins. For cell-based assays, our CRISPR cell lines offer that revolutionary approach to genetic manipulation, allowing precise and efficient genome editing. Our CRISPR cell line services are going to enable you to generate stable cell lines with precise modifications, such as gene knockouts, knock-ins, or even point mutations. These cell lines can serve as valuable tools for functional validation, target identification, and even lead optimization. The Cell Power Cell Line Development Service provides a solution of optimizing cell lines specifically for antibody production. So our scientists use advanced cell engineering techniques in order to enhance cell line performance, including increased productivity, improved protein quality, and enhanced stability. Our off-the-shelf stable cell lines are a valuable tool to antibody drug target validation as well. These are going to provide a consistent and reliable source of cells that express proteins at a known level, that this is then going to allow you to evaluate the function and identify potential drug candidates that selectively target the protein. Our immune checkpoint cell lines include PD-1, TIGIT, 4-1BB, and more. So after you've successfully identified your target and validated, purified, antibody candidates can now be generated for screening and optimization. We have a number of solutions in this area broken down into lead generation and identification, and then optimization of those leads. So to start off your lead generation, we can help you simplify your gene and plasmid design with our GenSmart intelligent platform. The GenSmart DNA construct design tool is free and it's easy to use. It's going to expedite the plasmid design process. You can select from two modes, either creating a construct or creating a library if batch design is needed. This can be done in just a few steps. And when you're complete, you can send that in for a quote, or you can place an order directly from the website. The codon optimization feature has additionally been upgraded to what we call our ZQ, and this incorporates deep learning in order to enhance the ability to express those difficult proteins that then results in a significant higher yield. As a pioneer and leader in gene synthesis, our service delivers high-quality DNA constructs tailored to specific antibody targets and optimization needs. The service facilitates the generation of diverse antibody variants, allowing you to screen and select from lead candidates with improved properties such as binding affinity, specificity, and stability. To expedite the process of DNA assembly and cloning, we do have a DNA assembly kit, and this kit employs a user-friendly one-step method that allows you to rapidly construct complex DNA libraries or variants for lead optimization. New to our gene synthesis services is our antibody gene to plasmid package. This is directly on our website and includes a sequence template in order to enter your variant light and heavy chain sequences. It supports multiple formats, including VHH, Fab, diabody, and more are in progress. Ultimately, the customer is going to receive expression-ready plasmids that are ready to go with their work. Our plasmid prep service is reliable and efficient solution. If you're looking for high quality plasmid DNA, this is going to be essential for that late stage lead generation and optimization in antibody drug development. With options for both research and industrial grade plasmid prep, we can cater to diverse needs in the field and guarantee that 100% sequence accuracy. If you're looking for a high throughput method for plasmid purification, our catalog products division offers automated solutions through their new AMAG Quattro. This is a modular design capable of purifying up to 24 samples with 6 samples per module, and all of these would be controlled from one control unit. Modules can be purchased individually or as a package. The automation provides that precise pipetting capability as well as prepackaged reagent consumables. The consumables are ready to go. You just place them in the instrument, and then the process is initiated using the easy-to-use interface on the control unit. You can expect yields of 300 micrograms to 1.5 milligrams of plasmid, and the plasmids will be low in endotoxin levels and can be expected to be completed in approximately 2 hours. We have an extensive platform options in order to design your antibody campaign. We offer the traditional mouse or rodent hybridoma platform with the option to add our monoboost immunization in order to expedite that lead generation. Additionally, we have our platforms based on positive cell enrichment and B cell screening or cloning. This includes our rabbit platform, MonoRab, which takes advantage of the unique rabbit immune system in order to generate sensitive and specific antibodies with little cross-reactivity. We also upgraded our hybridoma platform to offer plasma cell enrichment before fusion in order to enhance specific antibody leads. Both of these platforms have the option to add the beacon B cell screening to further expedite your screening timeline. We also have phage display libraries that support our single domain antibody development using alpacas. This includes access to naive and immunized libraries. And then if you're looking for fully human antibody discovery, we also have the option for transgenic mice. As you customize your campaigns, we do support a variety of immunogen types, including protein, peptides, DNA, and cell lines. And then to further expand on that previous slide, I wanted to provide a quick intro to our mRNA immunization that we launched last year. This particular immunization offering is going to help ensure proper expression of those difficult to express proteins like the GPCR membrane protein. Another instrument from our catalog products division includes a solution for protein and antibody purification. The AMAG Assay Plus is semi-automated instrument providing high throughput purification of up to 12 samples in less than an hour. And then the results of that is 80 mg of protein per sample. The AMAG process cuts down multiple steps compared to traditional methods, since you add the magnetic beads directly to the samples, and then the instrument takes care of washing and eluting. Our TurboChill platform is a proprietary protein expression platform developed by GenScript. It includes an option suitable for each stage of the development workflow with variables based on protein yield, production times, and deliverable characteristics. It's a scalable platform, so when you look at high throughput, this provides transient expression of 1 to 20 mils. It's our fastest at 5 business days, providing yields from 0.1 to 10 milligrams. This level is most complementary to earlier target discovery phase. Moving to our Express, this is also transient expression, but at a larger scale of 40 mils to 200 liters. Timeline is 10 to 14 days, providing yields from milligrams to grams. It's also capable of providing expression of diverse antibody formats, so single domain antibodies, bispecific T-cell engagers, bispecific antibodies across mouse, human, and rabbit species. Our high performance is our stable expression scaling up to 200 liters. The timeline here is going to be 6 to 7 weeks, providing yields ranging from grams to kilograms. This stage supports the later optimization goal in order to generate those larger, more stable expression volumes that are going to be needed to support your preclinical studies and IND. And then I do want to mention another option not listed here is our fuco-free expression in order to provide antibodies with little to no fucose. Since fucose can impact the immune response and other pathways, the antibodies expressed fuco-free are able to increase the antibody-dependent cell cytotoxicity outcome by enhancing the binding to immune cells like NKs. So if this is important to your immuno-oncology research, we can definitely provide fucose-free antibodies. And then, as mentioned early on, bispecifics are rapidly expanding the frontier of antibody drug discovery. Our GenScript scientists offer our expertise to support and guide bispecific development using that proprietary TurboCHO platform. So the capabilities incorporated into that are going to be specialized format design, AI-driven codon optimization, optimize production process, optimize plasmid ratio, multiple purification column options. So far we've completed over 700 different formats, or 70 different formats, I apologize. We've delivered over 5,000 projects and have optimized more than 6 development pathways. Examples of completion, you can see over here on the right. You can also find more on the website and use the interactive feature to learn more about specific format developability and manufacturability. And then to note, for any kind of specialty antibody services, we definitely have offerings in those categories as well, whether it's modification like conjugation and labeling, fluorescent labeling, we can definitely help out in that area as well. Lead generation and optimization in antibody drug development often involves the exploration of improved characteristics of the antibody. Our mutant libraries offer a comprehensive collection of pre-designed or custom designed variant libraries that cover a wide range of mutation types. These libraries provide the ability to screen large numbers of antibody variants to identify leads, to find those enhanced binding affinity, specificity, stability, and other desired properties. Our precision mutant libraries are powered by the GeneTitan platform that I mentioned earlier, enabling the improvement of antibody binding affinity via CDR engineering and antibody affinity maturation. And this can be done through saturation scanning, site saturation, and/or combinatorial mutagenesis in order to create a unique library. We can also assess the developability and functionality of an antibody as a package report. This optimization assessment includes humanization, optimizing to eliminate any kind of aggregation from developability. And then on the functionality side, we can assess affinity maturation and provide necessary FC engineering in order to enhance or reduce effector function. Humanization technology allows us to optimize antibody candidates for that enhanced efficacy and reduced immunogenicity. Once we have the antibody sequence, the processes are based on internal computational algorithms and molecular modeling to modify those non-human antibodies to make them more compatible with the human immune system while preserving their target binding characteristics. By minimizing potential immunogenicity risks, humanized antibodies have a higher likelihood of progressing through preclinical and clinical development stages. Just to refer back to those 2 examples I gave at the beginning, both those antibodies were humanized. So this service ultimately will generate up to 15 humanized monoclonal antibodies. Our scientists will validate using ELISA binding and EC50 affinity ranking. And then the top 5 would be provided to you, the customer, as a small-scale transfection in order to further validate. Having a confirmed antibody candidate brings us on to these next steps of preclinical evaluation. In this stage, many tests are performed according to FDA criteria to assess and collect data related to toxicity and pharmacogenetics. Non-human animal studies are also incorporated in order to determine whether that antibody lead performs as intended or whether it supports safe use. This section is divided into the candidate validation as well as IND study. And so here I want to highlight our preclinical grade plasmid prep that ensures that plasmid production is completed with the highest standards and extensive QC testing. This chart outlines what you can expect from the service for specific QC parameters and the method used. Some QC examples to note are sequence verification and DNA purification and confirmation. The ultimate goal is to ensure our plasmid prep service delivers you plasmid DNA with the highest purity, yield, and consistency, and again, is going to guarantee that 100% sequence accuracy. Anti-idiotype antibodies play a crucial role in the development and characterization of antibody drugs. They're designed to specifically recognize and bind to the unique variable region of another antibody known as the idiotype. These antibodies are generated using advanced recombinant antibody technology, ensuring high specificity and sensitivity. We also have a large collection of catalog anti-idiotype antibodies as well. These antibodies function to monitor the pharmacokinetics, immunogenicity, and efficacy of the antibody drug candidates in these preclinical models, and then give that valuable insight into the behavior and potential therapeutic impact. We provide 3 types of these antibodies: those involved in detecting free antibody through antigen blocking, the second detecting total antibody through non-blocking mechanism, and then the last detects bound antibody that is that is going to be the drug target complex specific. These can be combined with our custom ELISA kits that are powerful tools for quantitative analysis and conducting essential PK and ADA assays. We have a comprehensive selection of in vitro bioassays. There are 14 categories to choose from and over 40 ready-to-use assays for various targets. Among these include ADCC. This was mentioned earlier as part of the fugo-free antibody development. We also include immune checkpoint targeted assays. And as bispecifics have increased in development, we can also develop specific bioassays for your bispecific project. Our gene-to-lentivirus service enables efficient generation of high-quality lentiviral particles carrying the gene of interest, facilitating the functional analysis and in vitro validation of antibody drug candidates. The service begins with the cloning of the gene of interest into the lentiviral vector, which is optimized for efficient gene delivery. The lentiviral particles are then produced through a state-of-the-art packaging system, generating high-titer viral stocks. These lentiviral particles can then be used to transduce a wide range of cell types, including both dividing and non-dividing cells. For cell culture methods, we do offer cytokines and growth factors. These applications for this include, cell activity, functional assays, cell culture, and immunoassays. And then taking a look at GenScript ProBio and their chemistry, manufacturing, and controls, or CMC service, offers a comprehensive support for preclinical and clinical stage of antibody development. The service is specifically designed to address the critical aspects of biologics development, including that process development, manufacturing, and quality control, ensuring the smooth transition from preclinical research to clinical trials. During the preclinical stage, it's also crucial to have an ample supply of antibodies for your in vitro and in vivo studies, including functional characterization, efficacy evaluation, and safety assessment. So our large-scale antibody production service offers a scalable and customizable approach to meet the specific demands of the research, ensuring that high purity and biological activity. So whether it's monoclonal antibodies or recombinant antibody format, like a single chain variable fragment, our production capabilities can accommodate a diverse range of antibody formats. So this is the last stage of solutions related to your clinical development. I've mentioned our ProBio team already in a couple places and want to emphasize that ease of transition of technical transfer as the quality scale increases, because we do have our CDMO integrated into our processes. These final services that I share are provided by our highly experienced CDMO team. And this section would be divided into cell banking, manufacturer, and analytical services. So during the clinical stage, it's going to be crucial to have well-characterized and a scalable cell line that consistently produces the desired antibody with high yield and quality. The GMP cell line development and cell banking service cover various activities, including the cell line selection, genetic stability assessment, productivity optimization, and establishment of GMP-compliant master and working cell banks. ProBio follows strict quality control procedures and conducts comprehensive characterization and testing to ensure the safety, stability, and productivity of those cell lines. And lastly, the ProBio team offers pro-GMP manufacturing service. This is going to provide a seamless and compliant pathway for the production of antibody drugs for commercial use according to regulatory compliancy and cGMP requirements. Pro-GMP manufacture covers the whole process of solution preparation, cell culture, downstream chromatography and filtration, using a single-use technology in order to avoid any risk of cross-contamination. And then we're happy to offer support in any capacity, whether you are looking for a single solution, like expressing your recombinant antibody, or if you're looking to customize a project with multiple integrative services or products. I want to take this moment just to remind you of our 20 plus years of work and partnership. We've been cited in more than 74,000 publications and peer-reviewed journals. This is a little bit dated, but we do continue to grow in those number of products and services citations that we're offering. And then if you're curious what this looks like, it really just starts with a call to learn about what your project is and questions and needs you may have. The therapeutic antibody development process is complex and likely includes many projects within the scope of what you need. Each partnership is going to be unique and is going to be customized to each customer's needs. To do that, we do ensure that you have a dedicated project manager to your case that will be with you throughout the process in order to keep you informed, ensure and keep timelines, and make sure that you're getting the technical and ongoing support that you need. And that concludes my presentation, and I'm happy to take any questions.
Great. Well, thank you so much, Amanda, for your informative presentation. We will now start the live Q&A portion of the webinar. So if you have any questions you'd like to ask, please do so now. Let's go ahead and get started with our first question here. Can I use my own antigen to have you help develop an antibody?
Yes, absolutely. We can use antigen that you provide us, or we can take the sequence from you and we can express and purify it on our end. We can also help to optimize it to make sure that you're going to get the best outcome of your antibody.
Perfect. Thank you for that. Our next question is, how much protein can I purify from the purification instrument?
That's a great question. The AMAG assay will give you up to 80 mgs per sample, and then it can also hold 12 samples per instrument, so that's going to give you 80 mgs for each of those 12 samples.
Great, thank you for that. We have another one here. Can you humanize an antibody that started in alpaca?
Yes, absolutely. We can also humanize antibodies from mice and rabbit as well.
Okay, wonderful. Well, it looks like we only have time for one last question here. So we'll end here with this one. Can you help with the right format for my bispecific antibody targeting EGFR and CD3?
Yes, we can definitely help make recommendations as far as the best bispecific format. We can also help to optimize the sequence, expression, and purification process in order to get the best product possible.
Wonderful. Well, Amanda, thank you so much again for your time today and for your important research. Now, before we go, I'd like to thank the audience for joining us today and for your interesting questions. I'd also like to thank GenScript for sponsoring today's webinar. Questions we didn't have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. Just remember that this webcast can be viewed on demand for 1 year until April 30th, 2025. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Again, thanks, Amanda. And until next time, goodbye.